Um, then we are going to let you work more independently. So what we just wanted to do a little bit of a summary here towards the end about really what we've been trying to tell you is really how to set up a processing pipeline for analyzing single-celled RNA-seq data. And so we've tried to cover various different ways of how you can do things. And the truth about a lot of these is that there are still sort of open research questions, and there is no strong consensus in the field exactly about what is the best way of doing things. So, but here in this last chapter, we're going to tell you sort of what we think is the best thing to do, and the way that we typically go about when we try to process and analyze the data. So take this with a suitably large pinch of salt, and be prepared that other people might have different opinions. So as we were discussing, sort of this first bit here is really about before doing the experiment, and again, this is important for those of you who are bioinformaticians, that ideally you want to try to get involved in the project before people start pipetting and whatnot they're doing, so that you can have a say about what things they're considering and what parameters they're using when they're setting up the experiment. And hopefully that can improve your chances of being able to analyze the data in a meaningful manner. So we think what's probably the most important thing to consider here is the sort of the experimental setup in terms of batches, so that you avoid having a confounded experimental design, so that you're able to correctly address the issue of sort of technical variability versus biological variability, so you can identify and separate the two. Two tools that we've been talking about that are often quite useful for analyzing the data are UMIs, unique molecular identifiers, and spike-ins, typically from the ERCC, the External RNA Control Consortium. We are quite convinced that UMIs are very helpful, and that they in general improve the quality of your data and reduce the amount of noise. Spike-ins we're less certain about, and I think I speak for most people in the field that it's a little bit, question is a little bit up in the air about whether or not they are useful, and you can sort of end up spending, sort of sacrificing a significant fraction of your reads for something which may not be that informative at the end of the day. So that's something that you should think a lot more carefully about, whether or not you're going to be able to take advantage of the information that they provide, and if the information that they provide may even be helpful is a more important question. Another very important question in terms of the experimental design is how to balance the number of cells versus the number of reads. Typically, we're dealing with fixed budgets, and we have to decide how to split the two. What's happened now in recent years, right, is with these drop-seq protocols have made the costs per cell cost very low, which means that it's very easy to scale up to thousands or tens of thousands of cells without breaking your budget. But of course, there's no point in having lots of cells if you can't sample them very deeply. And again, exactly what trade-off to use here really depends on what question you're most interested in. Obviously, if your goal is to sort of try to characterize the diversity of cell types or try to identify rare cell types, then you want to go with the more shallow sequencing. Whereas if your main goal is to more trying to get an accurate estimate of sort of relative levels or absolute levels of expression of certain genes, in particular, sort of one is often interested in transcription factors, and they can often be quite lowly expressed, and then you will obviously want to have more deeply sequenced samples. Another aspect which I don't think is on here is what type of protocol to use. There are two main types, which is either the tag-based, which tend to be where, well, actually that's related to the UMI versus no UMI question. But there are these full-length transcripts which will give you try to cover the entire transcript versus the ones that are more 3' based. The latter are the ones that are compatible with UMIs, so hence they tend to be more popular. They obviously have the drawback that it's going to be very difficult to identify splice sites or different isoforms. So if that's what you're interested in, they're not going to be as useful. Then we finally have on the more computational side, we've already been talking about how to process reads. We recommend doing QC on the raw reads, typically using FastQC or CutAdapt. You need to map your reads to a reference genome. We think that STAR is the most reliable mapper, although if you are a very large data set, you probably want to go with Salmon. And then we have various methods for doing the quantification of the.
Sorry, you mentioned Callisto in the beginning?
Yes. Salmon and Callisto they do virtually the same thing and along virtually the same principles. So I think there are probably very small differences between the two.
Yeah, as far as I know, originally they did the same thing. Salmon has recently been updated to do a bit more extensive modeling of base frequency biases. So at the moment, Salmon is slightly better. Next month, Callisto might have done an update and Callisto will be slightly better. So they're essentially the same though.
So you need to quantify your genes, your transcripts, using these various different methods that we've covered. We think that feature counts works quite well. And obviously, if you do have UMIs, you will need to process them and take that information into account correctly. As we mentioned, this is a very, very hot and bleeding edge aspect of the field, and there isn't actually that much out to do there, many tools out there to do this, despite the fact that everyone seems to be using UMIs in their experiments these days. So expect to see quite a lot of development there in the next few months, I would predict. Further quality control, we think that Skater is great, not just because Davis is a very nice guy. It has a lot of built-in functionality, and it can allow you to remove cells or genes that are of dubious qualities for various reasons. You will need to do some sort of library size normalization. We think that Scran is the best available option that we have out there today. And depending on your setup, you may need to do some sort of batch correction, and we think that RUVS is probably the best option that we have there. Finally, we have 3 different types of analysis, which are quite common today. You're often interested in doing some sort of feature selection. We think that our M3 drop method does the best job there. Again, it really depends on what you're interested in. If you're interested in finding the genes with the highest variability, then you should obviously use HVG. But this is also something which is a very active area of research, and people are trying to really get their heads around what are the best features to extract and what should be the principles for doing so in a sort of unsupervised or even supervised manner in some cases. Clustering, we think that SC3 is a mighty fine tool to use there. For pseudotime, we have somewhat less experience ourselves, but we think that the diffusion map has sort of in particular, it has very nice theoretical properties, and it thinks to deliver very well in terms of the actual performance for data. So I think we will wrap up the main session here. And the last thing I need to tell you before you leave is that there is some sort of course evaluation for you to fill out. It's on your desktops. I might even be able to. Course survey. What happens if I click on this? Nothing. Oh, there you go. So please fill out the course survey before you leave. And I think that's it in terms of announcements. Yes, the exercise. We have additional exercises. So we have what we refer to as advanced exercises. There are distinctively no answers provided to these. And I think Vlad is going to explain a little bit more about that.
So those of you who have their own data sets, you can actually work on your own data sets and ask questions if you need. And those of you who don't have the data sets but are still interested in doing some analysis from the beginning, so we recommend to use this Conquer resource. So this is a very nice repository of data sets which were basically processed in the same way. So I think it's one of the groups in Sweden. They just took the publicly available data sets and processed them in the same pipeline. And they provide different reports. So there is a SCATE report. You can have a look at this. There is a QC report. And you can actually download the processed data set. So you can download the raw counts for any of the data sets that you're interested in. So if you basically decide to take one of these data sets now, download it to your computer, then what you can do, you can create a scatter object from this data set using this code here. So for example, I downloaded this data set from the website. I think by default it will download it to your desktop. So if you read it and then run these commands, it will create a scatter object where you will have the counts and you will also have TPMs, because TPMs are also provided in this repository. And then you can actually do all the steps that we've shown you so far, and basically try it yourself on this random data set. We haven't done it ourselves, but we would be very happy to see if you can do it. And there are some questions at the bottom of this page. This is just for the reference. If you want to follow them, you can, but you can also do what we've done with you in these 2 days. What else? I think that's it. So there is also a resource page in the course which contains multiple links. So these are the links to these different single cell protocols. This is the link to ERCCs that we showed you yesterday, where you can find all information about ERCCs and their sequences. Also, I added the Talula's GitHub repository link here. And also there is a nice list of single cell tools, which is basically under development. So it's being updated by different people, and a lot of scientists are involved. So I think this is the most updated list of tools available for single cell data analysis. So I think you won't find anything bigger list than this. And that's it. That was the last chapter. So if you go to this Concur website and download any of the data sets and then run the analysis on your computer, and then you can compare it with the report provided here on the same website. So you will see familiar plots, the same that we saw yesterday. Any questions?
Is there any useful discussion forum where I can get good answers for my questions in the single cell data?
So the question is there any discussion forums for single cell data analysis? I'm not aware of any specific. I think there is a Seek Answers forum, which has been used by a lot of people who are doing high throughput data analysis. Maybe there is a sub forum for single cell data. BioStars is another website where you can ask questions if you have any, and people will try to answer your question. BioStars. Yeah. Yeah, I think BioStars is specifically developed for this kind of questions. Yeah, so basically any question on bioinformatics. It's like Stack Overflow for bioinformatics, I would say.
Will the course material be kept on GitHub for a while?
Yes, it's been there for the last year, I think, and it will be there. So it will be updated. Next time we run the course will be spring 2017. So we probably update it just before the next time the course is run. Any more questions? And also the video actually were being recorded, and I think the video will be available after the course. So if you have anyone who wasn't able to attend the course, you can send them the links. I will put it on the course website, yeah, to the main page.